Uh, Welcome to the webinar, monthly webinar that we host. And this time our topic is uh, pushing the 505(b)(2) envelope. And we have here Dr. Patrick Maroon, who is going to be the expert who will be quizzed on different aspects of 505(b)(2). So it's a conversation that he and I would primarily have, and then the um, audience, you all can pose your questions as well. So let me take a few minutes to introduce the 505(b)(2) topic, and then we can delve into the discussion. So I'm assuming you can see me and hear me. Okay. This webinar is being recorded, and we invite your questions and comments. The questions will be, you can post the questions in the chat box, and I will try to moderate the questions with Dr. Maroon. And if you experience any technical difficulties, please reach out to Holly, and Holly's email address is provided on the screen. And then at the end of the webinar, we request you to do a quick poll of what was your experience with the webinar. Both Dr. Maroon's and my bonus are dependent on your feedback. So 505(b)(2), I'm just going to give you a very a big picture view of 505(b)(2). It is an NDA that allows for the approval of new drugs based on data not developed by the applicant. It is intended to streamline the drug approval process for drugs that are not entirely new but differ from a previously approved drug in some way. So it is still it's a new product. It's not generic, but it shares perhaps some aspects that can be borrowed from a previous or currently approved product. Now, here are some key aspects of 505(b)(2) pathway. Reliance on existing data allows the applicant to rely on existing clinical data from studies not conducted by the applicant. This includes published literature and the FDA's finding of safety and effectiveness for previously approved drug. You would need a right of reference, or use is the authority to rely upon and otherwise use an investigation for the purpose of obtaining approval of an NDA. Meaning, who do you get the right of reference from? You need to get the right of reference from the reference product holder. The type of changes that are allowed are typically seen for a 505(b)(2) application are going to be new dosage strength, dosage form, route of administration, formulation, dosing regimen, or even indication. It does not. I mean, the abbreviated new drug pathway is different. That type of approach is not applicable for 505(b)(2). Clinical studies may or may not be required. It depends on the situation, and if you work with an experienced drug development strategist, you would be able to come up with the most lean drug development pathway and find a way to negotiate with the regulatory authorities on the burden of proof. This is a phrase I've learned from Dr. Maroon that you need to bring to the table. There is a supernatural intervention here on the slide, so pardon me. The review process is similar to any other process. You need three things: safety, efficacy, and quality, and then there will be labeling negotiations. Label could be different from the reference product. So there is that flexibility possible. The advantages of 505(b)(2) is you can significantly reduce the time and cost associated with bringing a drug to the market, and it encourages innovation by allowing modifications and improvements to existing therapies. And the so before going into the discussion part of this web chat or regulatory briefs, let me briefly introduce Dr. Patrick Maroon. Patrick Maroon obtained his pharmacy degree from University of Pittsburgh and then a PhD from University of Florida. He joined FDA as a reviewer in the Division of Biopharmaceutics. In 1995, he became the team leader in clinical pharmacology and biopharmaceutics for the cardiorenal team. And in 2000, Dr. Maroon became the FDA regulatory science expert on in vivo in vitro correlations for modified release formulations. He was one of the very few scientists who pioneered the field of IVAVC and biopharmaceutics, and he was the man behind many policies, you know, around this topic. Then in 2008, he became the head of biopharmaceutics group in the Office of New Drug Quality, where he was responsible for all the review policy and research activities related to biopharmaceutics. In 2011, Dr. Maroon became an independent consultant for the pharmaceutical industry and then joined in AbbVie in the clinical pharmacology and pharmacometrics department. And currently, he is a senior research fellow within AbbVie, and personally, he has been a mentor, and I've learned, most of my basics in regulatory strategy from him. And Dr. Maroon, welcome to the regulatory briefs. We do this monthly, and today's topic is 505(b)(2), and I cannot think of a more eligible expert to speak on this topic. So please help us get a deep dive into this topic. So to start with, I want to set a stage for my first question, which is the Code of Federal Regulations, the law, describes the 505(b)(2) pathway, and specifically, I highlight this section B, where it says that the 505(b)(2) application may not be submitted under this section for a drug product whose only difference from a listed drug is that the extent to which its active ingredient is absorbed or otherwise made available to the site of action is less than that of the listed drug, and the rate, it's basically the same extent and rate, is unintentionally less than that of the listed drug. So I would ask you to please do two things. One, if you have any opening comments, please go ahead. And then two, if you can elaborate on the rationale for this requirement, because it's in the CFR, so that we all understand the thinking behind it. Please.
Yes. Thank you for the introduction. It was kind of you to really introduce me. The context for this requirement is that the intent of whoever wrote these regulations did not want for products that are not bioequivalent, but that would need to have the same exact label language as the innovator to go through that route. Which means if you don't want to change the label and you have no reason to change the rate and extent of absorption, then you cannot use that 505(b)(2) route. Which means, in other words, they didn't want any generics that is less bioavailable or any equivalent product that is similar to the listed drug to go to use that pathway for approval. You can still have a product that is different, less bioavailable in rate and extent, but it has to have some intention. Why did you do that? And for whatever reason you did that, and that's and that's what they mean by intentional. Sometimes you might want to slow down the absorption rate, or you want to decrease the Cmax, or you want to reduce even the dose that you want to give intentionally, And in that case, yes, you are allowed to use the 505b2 pathways, but the label will be different. The language and the label will be different, and I don't think you will be able to even get the AB rated rating, for that new drug. So that's, I think, the intent of that uh, uh, clause in the section in the CFR. It's very seldom used. I've never, in all my career at FDA, only saw it twice where it was an issue. But most of the times, most of the products that FDA gets, they change the rate of absorption intentionally due to some clinical considerations. So
I see. So I have a follow-up question, and then there is a question from the audience as well. Um, I, I I request the audience to write full questions because I am not a psychic, and the last time I checked, Dr. Maroom is not either. So you'll have to write a question that is self-explanatory, so we can have a reasonable discussion. So, Dr. Maroom, so let me give you a hypothetical example. So let us say there is a drug that is given twice a day, immediate release, but then the 505b2 sponsor considers an extended release to make it once a day. Naturally, um, your naturally you may not be able to match, for example, the C trough um, between the extended release product compared to the twice daily immediate release product. Would that be a challenge given that this law requires not less than the listed drug?
no, because the change is intentional, and if you're going to give it as once a day, for sure you do not expect the Cmax to be the same. It could be less available, less than the Cmax, or even the UC might be less. So in that case, the 505b2 is the perfect pathway to go for such a product. But I mean, you cannot approve the product just based on PK considerations only. You might need to have additional studies to justify that the difference that you see in the rate and extent are clinically acceptable from a safety and efficacy point of view.
Thank you. I hope.
And again, when you change from IR to ER, I don't think you can I mean, you have to have some studies to really characterize your formulation, as well as show that from a clinical point of view it is acceptable.
Thank you. I hope uh, Dr. Shivacharan and Dr. Manushree, your questions are answered with this discussion. Dr. Maroom, you brought up AB rating. So can I
Yes.
have a 505b2 product with where I can, pharmacists can switch?
Yes, I've seen a couple of times. It happens mostly for modified release formulations where, they were able to show that their test product, the 505, the new test product, is equivalent to another controlled release formulation that is approved on the market. So which means, and then in that case, they can get an AB rating to that approved modified release formulation. They will not be able to get an AB rating comparing to the IR formulation, but they will be able, for example, to get AB rating to another already approved, for example, modified release formulation. So yes, it's possible to get an AB rating. It depends on the type of product and whether you have a suitable reference that you could compare yourself to.
I see.
Yes. Yes.
So if the reference product is a solid dosage form and my 505b2, let us say, is a liquid, you're saying I may not be able to get AB rating.
Yes. Yes. I mean, a lot of times you also can get AB rating let's say you are using a different salt of your compared to the let's say the reference use one salt and the test use a different salt, and then so basically you cannot, you have to go through the NDA process to get that new salt approved. But in that case you don't need to do a full scale NDA. You can use the 505(b)(2) route to get your product approved, and many times they can show that the two salt forms of the active ingredient in the formulation that they use result in bioequivalent plasma levels. In that case they can petition and get the AB rating. Okay,
Okay. So we
so
have
that's
another.
one instance where you can use the AB, you can get AB rating.
Okay. So you're saying for AB rating, there are two requirements at least. One is the formulation should be basically
Bioequivalent.
similar formulations, the product basically. And then the second, the dosing regimen should be also
The
identical.
same,
Yeah.
identical, and the same route. You cannot get AB rating for a different route.
Okay. Thank you. Um, Dr. Ganesh Sangle, it's not clear what your scenario is. Please explain what that scenario is in your question. So, Dr. Marum, we have a question saying if we found a new indication for a phase 3 abandoned drug not approved by FDA, what is the way to approach such case?
Is it a new molecular entity that was never approved in the US?
It's not approved by FDA.
Yes, then you cannot use the 505(b)(2) scenario because there is no reference drugs that you can rely upon for information. So you have to go through the full route of the submission because it's considered to be a brand new molecular entity. Uh, unless the drug is approved in the US, then you can maybe go through the 505. But if it's never approved, it's a brand new drug basically. Even for like drugs that are approved in other countries but not approved in the US, they are still considered as unapproved drugs.
So, Dr. Marum, uh, to follow up that, why can't I use uh, the in this particular case about a drug that is, I mean, a drug is that's not approved by FDA yet, but maybe there are publications. So 505(b)(2) allows using to rely on publications.
Whether it's a 505(b)(2) or a full NDA, you can have what we call a paper NDA, where all the information is derived from literature. The only problem with such an application is a lot of times FDA would like to get the raw data to do their own analysis, and a lot of times this information is not available from the authors of the publication. But if it is possible to use literature data where you can get the source data from, so you just cannot just show a graph and say this is and rely on that and not have the source data behind that graph. But I've seen one, in fact, I remember I reviewed one drug that's completely based on papers, 100%, and we
Yes.
approved it.
Okay. Thank you. Um, so even if it is, to just summarize, even if it is the efficacy and safety was evaluated and published, the expectation is that the sponsor would have the underlying reports and data when they
Yes.
submit, like including bioanalytical reports and CFR, I mean the case report forms and such, right?
CSR, yeah. Don't forget, for any NDA to be approved, it has the clinical trial sites have to be inspected and they should pass inspection. So if it is based on a paper NDA and you don't have access to the source data, how are you going to allow FDA to really do the inspection? So there is some legal ramifications on the use of these paper NDAs, and that's why most of the times many companies opt to repeat the same studies. It's easier and more straightforward for them to just
Okay.
rely on their own data. But it's possible to do paper NDAs, yes.
Okay, thank you. Um, the next question from the audience is that what kind of a bioequivalent studies are to be done if we want to develop liquid oral dosage form for approved solid oral dosage form?
I mean, a simple BE study could
Yeah,
suffice. And you would need also a food effect study.
yeah,
So a simple PE study with a food effect study will be okay. And in that case, you can use the 505(b)(2) route. That's
yeah.
the ideal case where.
Okay, thank you. And the next question is, can 505B2 route be used for a new topical combination of two already approved FDA product, I mean, FDA approved
Yes,
product?
absolutely. Yes, because both of them are approved, and you could rely on the information from these two applications to put your application together. In that case, I don't think only you can base it on BE. I think you need to have additional information. For example, like the drug interaction between the two drugs, and maybe you might need also, because of the combination, you might need to satisfy the drug combination requirements. So, but yes, it will be a 505(b)(2) application also.
Given, I mean, from the question, it's not clear of what the approved uh, products are, the route of administration. But if the topical is new, then wouldn't you say, Dr. Maroon, they may also need to do some tox studies?
It depends on the dose that they're using. If it is within the doses that are
No,
already.
I mean saying from you changed it from, let's say,
Also
some other
topical, so you
formulation
might
to,
you don't need to repeat all the tox studies. You might need to repeat or to do some additional tox studies because of the route of administration. But the initial tox studies about how you can go without any problems in terms of toxicity, you can get away without repeating them. Yes, you might have to do some partial studies, but not all the. You don't have to repeat all the studies.
okay. For fixed dose combinations, what
Hmm.
if the indication is different from the one from one of the individual components?
So you want to basically develop a new indication. If the indication could be supported by a PKPD study, you could use that route. Otherwise, if you could not have a PKPD relationship, or it's not known, then you need to do a clinical trial to show that your combination is safe and effective. And combination products really sometimes are tricky because the doses that are used are different than the monotherapies. So you might need to have some dose ranging studies or some additional information to justify your choice of doses for the fixed dose combination. Oh.
Okay, thank you. And then I'll move on and then I'll come back. There are a lot of questions coming from the audience, so I'll come back to those
Okay.
in a minute.
Sure.
Um,
Sure.
so, are efficacy and safety studies always required? Um, uh, you and I both are familiar with the word required to
Yeah.
file a 505(b)(2) application.
No. It depends on the type of product and the type of application that you're using. I mean, the type of indication that you are intending to have. But you can have uh, 505(b)(2)s only relying on PK. I've seen cases where only they repeat the tox studies, and sometimes they do one clinical study, and they add some supporting information like a PKPD study. And sometimes I've seen cases where they have to do two studies. So it depends on the situation, but it is not always required to have a clinical study in terms of safety and efficacy.
Thank you.
I
Um,
give you an example of a salt. Another salt of a product can be just approved based on bioequivalency results, without any other information.
I see. So one of the questions from the participants: can 505(b)(2) filing be based on PBPK modeling and get a bio waiver?
By the way, for what? For what?
It's not clear, but I'm assuming it's for the bioequivalence trial.
No, I mean you have to have some data on the product. You cannot approve a product with zero data. It's not like you can use virtual bioequivalency and PKPD relationships, and rely on model informed drug development. But you have to have a minimum set of data to show that, look, we have some human experience. You cannot just have a completely virtual NDA, I would say, whether it's PKPD or PBK or whatever tool you use. So yes, you have to have some data. I mean, the PPK model or the PKPD model will really save you many additional studies, but you still have to have some data. You have to basically get plasma levels on the product that you're trying to market. It's a CFR requirement.
Yeah. Okay. Thank you very much. And to probe that there is a great push, I have seen, Dr. Maroon, especially outside of the US, regulatory authorities are pushing this use of PBPK, and they even come up with some new terms, PBBM, and but physiologic
Mm.
models pretty much, in lieu of in vivo bioequivalence trials. Can you
Yes.
generally opine on that practice?
Yes, I mean, recently, FDA and some other regulatory agencies are allowing the use of modeling and simulation in lieu of actual studies. In fact, this concept is not really a new concept. Don't forget the IVIVC guidance was published in 1997, where it allows you to use the correlation that you establish between the solution and plasma levels to get a waiver for a study or to get approval of a new strand. So the concept is not really a new concept. It's been there and it's been used for many, many years. What is new is that now we have an additional tool, which is a PPK model that will allow you to predict the plasma levels and compare the test versus reference. And that's where it's taking it's becoming more and more common. But the problem with PPK model is a lot of times when we develop the model, many people use a very permissive acceptance criteria. And for you to be able to use it for bioequivalency purposes, the model has to really meet tight criteria. you know. So this classical twofold acceptance criteria for PPK modeling will not be viable or will not be acceptable. So you really, I mean, yes, you start with a kind of initial model, and as you accumulate more data, you basically will refine the model, and it becomes much more predictive with greater accuracy and precision. If your predictions are very bad or very big, then I don't think you will be able to use PPK modeling for virtual bioequivalence. So the model has really to be very, very good, before you could use it. And that's where the challenge is, to come
Yeah,
up with a model that is very tight.
that's a good point. Because for bioequivalence, you're trying to pick up, 5%,
5,
10%,
10%, yes, exactly.
differences. So your model has to have the capability to, render that,
Yeah.
to differentiate between false positives versus true positives.
Yes. And also another component that people do not really pay attention to, because you are using your dissolution as your input function. So, which means if you don't have a good dissolution method, you will not be able to really predict or pick up differences among different formulations. So, one other requirement that really people kind of miss is that you have to have a good distribution method that's discriminating, that is able to pick up differences among formulations. If you are not able to do that, then same like the IVIVC, you will not be able to be successful in using this virtual bioequivalency tool.
Okay. So a question from the audience. I am kind of paraphrasing it a little bit because it's not clear. I think the question is, if there is the reference drug is oral,
Mhm.
and then product, to be specific, oral product,
Mhm.
and then the 505B2 is a transdermal.
Mhm.
Do we need to evaluate efficacy?
I would say yes, unless you have a PKPD model developed with the transdermal route, and you could, use it. But if and very seldom, I think you have something like that. So my question is, most probably, at least for the first product, the first transdermal product, you would need safety and efficacy. Then the second one that comes in, if they can show that they can produce equivalent plasma levels in terms of Cmax and EUC, then in that case they could maybe rely on exposure measures to show that they are equivalent, and then they don't need to do a efficacy study. But my feel or my opinion is, for the very first one, I don't think you can get away without having to do at least one clinical trial.
Thank you. There is a comment, so I'm just going to read it.
Okay.
I guess we don't have to react to that. Mini pig dermal tox studies would be required in the case of drug combination 505B2. Duration will depend on the clinical use. We can say sure.
Yes, we need to have yes. It's a
Yeah,
different route, but
yeah.
like I
So
said before, you don't need to repeat all the tox studies, only some of them.
right. So this goes back to the previous one we talked about for dermal.
Yes.
Maybe,
Yes.
so thank you, Dr. Jadhav, for that quick comment. The next question is, if FDA recommendation has proposed fasting and fed study, do we need to perform both the studies against listed drug or
Okay.
food effect?
Yes. It
Hang on,
depends on the label.
hang on.
It
So,
depends on the label. Sorry, I interrupted you.
no, I just want to complete the question so you understand it.
Okay.
So are the food effect study be done against the test product?
Okay. Okay. It depends on the indication. So what happens? There are two cases here. One, if the drug is always given without food or irrespective of food, then you need to do a fasting BE study and include a food effect arm to show, or your test to kind of quantify the food effect of your product. If the drug is going to be given always with food, then you would do a fed BE study and include a fasting arm. I mean, it's clearly written in the bioequivalency or the food effect guidance of FDA. Now in Europe, and for generics, it's a little bit different. In Europe, if the drug is given to always with food, or if you have a modified release formulation, they would require you to show bioequivalency under fed and fasting conditions. So it depends on the country and the regulatory agency, but FDA is a little bit different, as I outlined.
Thank you. So there is a question. I'm going to still paraphrase and guess a little bit. So, what are the pediatric study requirements for 505B2 products?
It's the same as a new drug. It's no different.
Okay. So, I'm not sure what the motivation is for that question. I guess
Un-
the the uh, the participant is thinking, if the original the listed drug is not approved in pediatrics, or they did not conduct pediatric studies.
It
What
doesn't matter.
happens
You still
to
have
the,
Yeah, it doesn't matter. You still have to meet the
yeah,
the requirements. A lot of times There are very, very old drugs, even they don't even have clinical trials that are on the market.
Right.
So basically, what you have to really meet the current standards. Now, unless your product would never be used in pediatrics, then you can get a waiver, or for example, sometimes you can get a deferral, or you get more time because, for example, the patient population is very small and it's very difficult to recruit people in time to meet the requirement. Then you meet with the agency and kind of come up with an agreement with them, you know. But it is no different than
Yeah.
any NDA or.
Okay.
And it doesn't matter whether the reference drug has the pediatric or not. If it doesn't have, you have to meet it, you know, as
Yeah.
a test.
Yeah. So, in other words, is it fair to say if you are developing if you are choosing a 505(b)(2) pathway today, you will have to play by the rules of 2024, not
Exactly.
1924.
Not 1913, yeah, not 1924. Yeah.
Okay. Um, okay. And then is 505(b)(2) only for small molecules?
No, you can do, but the biologics have a completely different pathway, so all this doesn't apply to them. They have their own, pathway, which is a BLA, not an NDA. So, and the regulations really do not address biologics, but the concepts could still be used. You know, so regulation-wise, we don't have regulations, but the same approach and the same thinking could be used. I think it's much more complex because there are many, many different issues, you know, that you have to deal with biologics that you don't need to deal with in terms of small molecules.
Yeah. But can you not say that now with biologics, you're more, closer to the biosimilar concepts?
Yes, exactly. I mean, the biosimilar, if you like to think about it in the analogy, is a biosimilar application is somewhat similar to a 505(b)(2) application,
Yeah,
even though you don't call it that way, you know, because
that's
it
right.
doesn't fall in that category.
Yeah, I agree. I just want to share a thought, historical thought, which is before the advent of biosimilars,
Mhm.
insulin products were approved under 505(b)(2), although it's a biologic, because there was no other regulatory pathway, and so FDA used to uh, accept uh, quotes biosimilars that was not available, or the
Yes,
word was not
yes,
available at that time, concept under 505(b)(2). Yeah.
yes. I mean, not only insulin, but I mean, you have a lot of, for example, a lot of products that are of animal origin. For example, for
Tamarin.
example, we have a drug, creon, which is a mixture of enzymes that help you digest, and they are made from, they are extracted from animals. You know, this is technically not a new. It's not a drug. It's a biologic. You know, so that case, you know, it is a biologic, but we deal with it as a drug. You know, another case, for example, we have armored thyroid. Another product that we have is extracted from pig glands. You know, again, it is a drug, but technically it's not a drug. It's a biologic, but it's reviewed somewhat as a drug, and you know, it has its own issues. You know, but the rule does not fit to everything in biosimilar in biologics. You have to look at case by case, the indication, the complexity of manufacturing, antibodies, antibody drug conjugates, all these exotic biologics really have many issues that I don't think you will be easily able to really meet all the requirements, even for a new drug. You know, so.
All right. So switching gears a little bit in terms of the regulatory strategy, uh, do you recommend, Dr. Maroon, that 505(b)(2) sponsors uh, avail pre-IND meeting pathway?
My opinion is that the sooner you meet with the agency, the better off you are. Now, whether you absolutely need the pre-IND or you can do it immediately after the IND would be still give you the same answers. It's just a question of when you are ready. Sometimes you might need to open an IND to, for example, test different formulations of your different of your platform or your drug dosage drug delivery platform, and then you need to have some idea before you go to to uh, to FDA, for example. So you need to open the IND and then meet with them. Other cases where everything is straightforward. Uh, I mean, the more you meet with the agency and the earlier you meet, the better off you would be, I would say.
Okay, so in other words, that if I am a 505(b)(2) sponsor and then I have a clean slate, maybe I have some in
Mm.
vitro uh, experiments done and maybe an animal study. Should I consider going to meet with the FDA for a pre-IND to discuss the registration pathway, meaning the
Yes.
type of studies that I intend to submit with the NDA, and if that would get me approval?
Yes, I think sometimes, for example, if you're using different doses or different excipients that are kind of novel, and you need to really do some studies before you even open your IND, I think it's advisable or recommended that you would meet with FDA to see whether you have all the information that's required to open the IND. Sometimes you might be surprised that there might be some recurrence that you did not think of that FDA would like to see. So yes, I mean I would say uh, uh, again depends on the situation, but yes, I would say always try to meet with FDA as early as possible.
Yeah. So the advice that I usually um, uh, give when I'm working with some sponsors is that it is important to have a little bit matured data about your product before you can propose, especially I mean, if you're proposing a full-fledged efficacy safety evaluation and so on and so forth, perhaps it's okay to meet upfront. But if you are proposing some kind of innovative approach for a accelerated or innovative, I would say, registration pathway, then meeting without some data could actually be detrimental in the negotiations. What would you say, Dr. Maram?
Again, it depends on the situation.
Yes, yes,
I mean, yes, and in this case, you're absolutely right. But in other cases, very simple. Like you just change the salt. I don't think there's anything to discuss. You just
that's true.
do it, and it's very straightforward.
Yeah,
Or if you're trying to get a second modified release formulation on the market that is equivalent to already existing modified release formulation. In this case, the pathway is much easier. Now, if you want to get something novel that was never tested before, I mean, one meeting only, alone is not enough. You might need multiple meetings with different even people within FDA. So I agree with you.
okay. So the next question is how to support a different PK shape for the 505(b)(2) product compared to reference. So we could
I
take,
mean,
sorry, go ahead.
yes, in that case, the model informed drug development is a very good, pathway to follow if you have a model that takes into consideration or that can explain the differences in shape. Uh, that will be a long way of helping you explain these differences. But you need really to design a specific study where different input rates are used, for example, that results in different PK shapes, and try to develop a model with these that you could say, then you could leverage with FDA to say that, look, I understand the impact of having a different shape on the impact of my safety and efficacy. If you're not able to do that, then the only way you're going to really explain the PK shape is by having a clinical trial.
To add to that, you can think about innovative approaches in terms of shall I say, lessening the burden of uh, bridging a different shape to for the 505(b)(2) product compared to the listed drug, and um, you know especially if there are other shall I say extended release products approved, then it makes it a bit easier to make a case. If there are different shapes that are already approved, compared to there is only immediate release, and now you have to change the shape quite drastically, because you will never be able to meet the shape of a BID using a QD regimen. So that is going to be where I think MIDD can really help. And for some of the indications, like you said, it may be not practical to do clinical efficacy studies under various release characteristics. So that is, I think, one of the, uh, I want to say, most critical aspects of 505(b)(2), where the company, I would say, should work with somebody really experienced in this field, uh, so that they don't have to do unnecessary clinical testing. There's a question here saying, if the listed drugs does not have pediatric indication, but a 505(b)(2) product wants to add a pediatric indication, how can this be taken forward via 505(b)(2) pathway?
I mean, they have to do the dedicated studies to support the pediatric
Right.
indication. I mean, even whether they want it or not, they're required to do it. It's like they have no choice, I would say, unless they can get a waiver or can demonstrate that this drug will never be used in a pediatric population. Or some ages might be waived,
No,
maybe
no,
not
but
all
they
ages.
already are intending to get a pediatric indication, which means I'm reading that it is possible that there is.
Yes, then they have to do the pediatric program, and they have to meet with FDA and discuss the program, the proposal, and get approval from FDA about their plan, and the design of the study, the endpoints, all this. It's
Yeah,
basically a full-fledged pediatric development program.
yeah. Can you comment on? I know this is not. I'm just uh, following up on this discussion. So, if I'm a 505(b)(2) applicant, and the original uh, listed drug doesn't have a pediatric indication, but I pursued it and I got it, do I have any implications on exclusivity?
Yes, because you did the study, so you get the exclusivity for that product, not the other product. So if somebody wants to, so they can only use your product in pediatric patients. They cannot use now. They can use it off label, but technically, from a regulatory point of view, the pediatric indication only is applicable to to the product that did the studies.
Um, I can also uh, announce that the next regulatory brief is going to be um, on extrapolating efficacy to pediatrics. We have a, a, a very experienced expert, Dr. Jing Liu, who is a senior director of clinical pharmacology at Pfizer, and she will be having this kind of a conversation with Dr. Joan Court Bradley on August 28th, a similar setup. So if you are, and I have seen some of the questions are about pediatrics, and also there is a great push to having pediatrics approved sooner than later from a public health perspective. So this topic is timely, and we would hope to see you again on August 28th noon. So we have one more question here, Dr. Maroon. If a pro-drug is developed for existing drug, again, I'm trying to read between the lines here. So there is a, I'm assuming that the participant meant there is a drug product approved now, and then the 505(b)(2) sponsor developed a pro-drug for that drug.
Yeah, that's a new. No, you cannot go to the 505(b)(2) because it's considered a new molecular entity.
Okay, thank you. You can register for the next webinar by taking a picture of this QR code, and then you can register. And please, if you like it, you can send it to your colleagues as well. All right, that brings us to the end of this webinar. Thank you very much for joining us today, and thank you, Dr. Maroon. Have a good day.
My pleasure. My pleasure. Thank you.